next presentation we'll call Gemma. It's binary. It's a hemostatic device. As you understand, it's something which stops the bleed. And why we would bring this, you can see it right now, right? It's all good? Richard?
Yes.
Okay, all good, right?
Yeah, you're good.
Okay, great. So we already spoke about our team. And here's the situation we have in our daily lives, military, not only military, it happens, could happen anytime, injuries. So acute bleeding and consequences. It just statistically, on the Second World War, it's up to present time, we have cause of death was 60% wounded on the battlefield because of bleed. And based on military time, in the magazine, 90% of death occurred of the soldiers because they couldn't reach injuries and they couldn't reach medical facility was the bleed again. And this is just a quick example, kind of it's little showed to you on the visionary how it happens, the blast, the injury, the bleed, and the vessels. They use product, you can see it's Silox, it's current actually product used in the military. And this is the example of what they use currently in military, medical field, emergency rooms, Silox, Quick Clot, and other products, way more, and then trauma centers, and so on. And why did we come up to this area? Well, these current hemostatic compositions are, first of all, how they structure it, what they consist of. Hemostatic agent, the one they use, it's natural polymers, which is great, based on chitosan, alginic acid, kaolin, zeolite, some of them based on those type of products, and different components. Chitosan, as you know, it's a biopolymer of actually the shellfish skeleton of the shellfish, crab, lobster. Well, not using directly the shellfish, the processing and chemically producing the powder form. And alginic acid, it's come from the algae. It's actually seaweed. And they found out by using this molecules are the cause of blood stopping phenomena. And kaolin, it's mineral, comes from aluminum silicate, and it's part of a group of industrial minerals. And zeolite, it's come from lava rock, and that's the granules they use as substances based on the zeolite they use to stop the bleed. But there's a downsides behind it. Even being a natural product, they have a downsides which they are not absorbable. They're loose granules. They have to be placed in the wound or in the bleeding area, and they cannot keep it there just without supervision. And certain time could be 2 hours, 3 hours, no more than that. At times, you have to be quick to be removed, to be washed out. If they don't, that's what we have. We have an exothermic effect at times, could happen right away or later. What that means, some of those granules could cause the burn of the wound tissue and damage them. They help us stop with the bleed, and then might damage on top of it. Some chitosan-based product, I mean, they are non-absorbable, most of them, and they may lead to necrosis, meaning tissue damage, and causing decrease the blood supply to that area. A lot of just stops too hard on the pressure area on the wounds. And engorgement effect, that's where they put a lot of pressure and very firm. And if you don't remove that substance, that powder, already become like a hard rock consistency, it can damage surrounding tissue. It's helped, person survives, but if you don't remove them on timely basis, it may bring another damage to the tissue and the person. TRIZ in pharmaceutical, we spoke about it in a previous presentation, and here we are as the company who are only company who are applying TRIZ in pharmaceutical and healthcare industry. And we have to have a hypothesis to you have to figure it out what's the next for the new hemostatic. How's that should be look like? What should be different compared what the existings are? First of all, it must quickly stop bleeding faster than existing, because some of the bleeds coming from large vessels, and every minute counts. It has to cause a firm pressure on the bleeding site and without consequent on surrounding tissue, not causing necrosis or over pressure to the body and to the wound. It should be used in small amount and should be able basically cover as different size of the wounds. And explain to you later, actually, small amount, or right now why it's important, because it's a bigger wound. You would assume you have to put bigger amount to it. That's what they do right now. They have to cover the entire wound. But just imagine carrying all the supplies with you on the battlefield, or carrying supply with you when this in the injuries happens or some emergencies. It's problematic. So we need to have a smaller amount, but that smaller amount could be able to expand and cover the wound size. And new hemostatic has to be not immunologically rejected, too. It's not to be rejected by the body as a foreign or being allergic reaction or any means or causing the burns, and should have capability resorption, meaning metabolite. When you put that powder or granules, whatever you put in that wound, should get degradable, biodegradable, not to stay in the wound. That means you don't have to yank it out, that powder or granules. It should be body friendly and tissue friendly. And after applying to the bleeds inside, after the job is done, no debridement needed. You have to leave it alone, and it works for the job it does, and it doesn't need to be removed. So prism of TRIZ again, we had the problems. We need to solve it, find the solution. We utilize this prism of TRIZ, and we come up to a solution. Contradictions. All right. First TRIZ contradiction. From one side, a new substance must be quickly stop the bleed. And tamponade must be tight and firm enough to stop that bleed. On the other hand, rapid and firm pressure on the bleeding side may cause ischemia and narrowing of the vessels, that's what they're pressing on, and cause the damage to the tissue, that's what pressed on. So we have to avoid that tamponade must be firm, same time not firm enough. The second contradiction of the TRIZ. On the one hand, in order to stop severe bleed from multiple injuries, we would need to use a large amount of hemostatic substances, or larger wounds needs bigger, because of the volume of the wound is equal to the volume of the powder placed in the wound. On the other hand, it's difficult to carry, I just mentioned before, right? This large amount on any place we go on a regular basis, or battlefield, anywhere we have to be just in case the injury happens. So it has to be a small amount and cover the wound and do the job as far as stopping the bleed. And hemostatic substance should be in small amount, as it says here, and at the same time cover the large wound here. The principle to solve problems. The principle of rejection and regeneration. It's briefly defined as when we fulfill the purpose of becoming an unnecessary part, an object must be discarded after we use that part, right? In this case, it means the complete biodegradation of artificial thrombus, because we call this artificial because we're putting from a site. It's not your own personal thrombus, or its parts after the function has been performed. Even then, fragments of an artificial thrombus will remain in the wound because we were looking for a biodegradable product. The principle of solve this problem, it's a principle of self-organization and principle of dynamism. In self-organization, in this self-assembly, active substance from inactive precursors, a gel-like implant formed from two dry components in the mixture of the present blood. The moment our product touches the blood, it creates gel-like composition. The terminal fragments of these components are firmly attached to the wound. It attracts to the tissue and edges of the blood vessels, it tightening them, and that's what's preventing them from rebleeding. Why use that word rebleeding? Because after it dissolves or biodegradability, it might be rebleed, but it's not. So we have to look for this component as well. In principle of dynamism, this principle states that the characteristics of the object should be changed, and that to be optimal at each stage of work when it's function. In our case, that means that different sizes of the wounds, the same dose of the drugs or same amount of the powder form should be held for different sizes of the wounds, and only the percentage absorbed of blood would change dynamically. Meaning, it depends on the amount of the blood. It's like a sponge, like going to be absorbed to that same amount of powder, but different amount of the blood based on the bleed. That's what the dynamicity stands for here. Well, this is a little more here where the research we've done. It's more overseas. We tested acute bleeding on in vivo. It was done on animal-based studies, and here we come with that product called Gemma. This is quite quick excursus of current existing biodegradable polymers. It's already existing on the market. It's some polymers which are biodegradable. It's just example what's we have on the existing chemicals, but not what we're going to use. Stages of research and novel hemostatics. Here it comes. We want to select polymer with high adhesive properties to vascular endothelium. That's the clue here, one of the clues. So it has to be attracted right to the vessels, not to entire wound, on a specific area where the bleed is. For pharmacological study, different compounds. We design this in vitro interaction with hemostats and bleeding vessels. And the recording rate of how the polymers are swelling and what's the time necessary to formation, the clot formation. And we have to record the time necessary for clot dissolving afterwards without recurrence of bleed. And discover the best candidate, of course, for the hemostatic is composition, right? For the major bleeding. And we did studies on rats, rabbits, and pigs. It was extensive studies with great outcome. Here's the hemostatic our device, and we're looking for its existing enzymatic degradation mechanism. It's one is cleavage cross-links, mechanism two, transformation of side chains, and cleavage of backbones. Our product, it's more in this area, that combination that we have working on the three mechanism of cleavage of backbones. We also, by discovering a hemostatic device, Gemma, we use pharmaceutical acceptable salts. You can see from there, it could be the chlorides, hydrobromides, phosphates, sulfates, and other salts, organic salts, and pharmaceutical acceptable bases, which could be formed. These bases could be formed with Gemma, given together in different concentrations. And we have Gemma as a powder form, composition, and component with different excipients, which are FDA-approved generic drugs. We combine them in certain dosages and different combinations come up with the different products. Gemma components are used as therapeutic drugs, yes, for something else, was approved, but this is called repurposing them for unique, to use them for unique purposes. mechanism of action based on mechanical wound tamponade when it's pressure on the wound, with the formation of gel-like biocompatible, meaning not causing any allergies or any damages to the surrounding tissue. The thrombus, unlike the natural one, is formed in less than 3 minutes. It's much faster than existing hemostatic devices. So we came up with two products, K1, K2. That was quite a discovery. This Y2 you will hear in a minute. Again, using mathematical modeling and TRIZ principles. We came up with composition K1, K2. They are both are hemostatic composition binary, have high adhesive properties to vascular endothelium, great attraction to the bleeding site. They are both are biodegradable, they're water soluble, and they are, if they do the job and some leftover or needs to be evacuated earlier, or they just washed out with the water or normal saline without side effects. In vivo studies, just gonna mention to you before we go to the graphs, and we use the Silex. We use the one used in U.S. Army and military currently, and we use Gemma, K1, K2's control group. We use no therapies at all. In control groups, we had 6 rats. Each animal had, so the procedure we had done was under anesthesia, of course. Did some cuts, wounds, and unfortunately that control group has to gonna die within certain timing, 35 to 40 minutes. Experimental groups, which are Silex and Gemma, K1, K2s, we use 7 animals for each groups, and no animal died in that experimental groups. And bleeding stopped overall, and it's K1, K2 similar within 3 minutes compared to Silex 18 minutes. You could make your math how many times faster our product works than existing Hemostatic products. And it's all statistical truthworthy. The bleeding time. We use larger organs like the liver, heavy blood supplies, organs, liver, spleen, and after applying Gemma, the injury was decreased, and within 89-92% compared to control group with similar injuries. Bleeding time. Shortening bleeding time for experimental, like spleen injury I just mentioned as well, was observed with Gemma. Now, here we come with K1, K2. What's the difference in, we're talking about wound tissue regeneration. So as we mentioned before, K1, K2, both of them are great on stopping bleeding, timing, and functionality. When it comes, we noticed on experiment that when we carried out on the rats, we noticed that first group K1 and second K2 were applied in each group. This, of course, was combined with, compared with Silex. And same study we did, we noticed about wound healing timing were different compared to K1 and K2. Silex wasn't much any competition in that part. Now we're talking about these two K1, K2 Gemma products. The one which had been treated with Gemma K2 were healed twice as fast, whereas a control sample Silex, of course, yeah, we said that, right? Wound healing efficacy. And we did not differ from the control group even with Silex. And epithelialization and wound regrow with K2 group was initiated on the second day and much faster. So, and it was statistically confirmed on the K2, a Gemma K2. And this is just quick, like calculation time based on central bleeding. We done on the central arterial bleeding, on the skin, spleen, liver. This is this studied product, and you can see with the control group, the bleeding just continues exponentially until animal dies. Here is Silex, doesn't stop as much. It does stop bleeding, but not as good as about these two products, K1 and K2. And this is sorption. Remember I mentioned to you about this sponge effect, right? Small amount for bigger, for different size of wounds, or based, or rather say, it's not even size of the wounds matter, of amount of the blood or bleeding site. So the more blood comes out, our product was absorbing way better, and exponential absorption. You can see here K1, K2 practically similar. So Silex was absorbing blood not as good as our products, 10, 20, 30, a lot of times better, way better than existing product on absorbing on the blood. And control is control, not much effect. This is, we're talking about wound area healing, where we mentioned about K2. If you notice here, K2 start day 3, day 9 already healing process faster. K1 also does some effect. It actually doesn't prevent, doesn't cause problem from healing, but K2 was even exponentially increasing healing process compared to K1. It's different composition in those two Gemma products. Well, we complete the composition studies, as you've seen, and now we have a great results data, and we applying for Gemma. It is medical device. It's not the medication. It's not the drugs. It's all for external use, and it goes for clearance for FDA 510K clearance in progress as we speak. And this is just, I combined those slides as far as Gemma and Silex all together as far as their bleeding time, how fast Gemma stops, our product stops the bleeding, and how great absorption effect of our product versus Silex, of the blood absorption effect, the absorption effect. Advantages. Well, definitely it's biocompatible to the body tissue. It doesn't cause side effects that current products do. It's no tissue necrosis observed, no systemic or local allergies or reaction were observed, no burns effect on tissue or any crystallization on the wound area. It's absorbed 70 times even more blood than existing products when it comes to the absorption time, absorption effect. It is biodegradable, no wound debridement, no interference with wound healing. Gemma-based, it's actually an inexpensive material. That's important as well. It's natural semi-synthetic polymers based. The manufacturing and sterilization, it's simple. It's simple, and there's no need. It's not affected by heavy sterilization. It's functional after sterilization, and it's could be used in different type of products if it comes to the market and the packaging process. And the market opportunity, definitely it's ongoing. Still, there's a growing need for it. And we looking for as a startup company for investors, for collaboration, now for the grants applications. And that Gemma could be used in multiple fields. And where those fields are, so it's good: military, hospitals, trauma centers, acute bleeding injuries, surgical department, ambulance, first responder, police department, firefighters, and a part of the first emergency kit for general population. Have it in your car or at home. It's small samples, just it's life unpredictable, things happens. And we have a absolutely IP behind it, intellectual property for this matter. For our, as you can see here, again, creation of new medical drugs based on TRIZ and computer mathematical modeling. That's what we had one of the publications. And our NOIGEL team, as usual, ready for the next questions, or Dr. Farber had some comments to make as well. So we'd like to go for the next level. Richard, any questions? I'm glad to answer for your questions. Could be for the previous presentation and polymyxin and this one called Gemma, hemostatic device.
Okay, small announcement. Can you hear me?
Yes.
Great. So I just had a meeting with our team and with our patent attorney, USPTO, and good news, Dr. Kleyn, that high probability that for previous presentation polymyxin, our patent will be issued in a few months.
Great.
We almost won.
That's right.
Take many years, but this is true.
Thank you. Great news for all of us in the healthcare industry. Thank you.
Sometimes patent takes years. For cancer, it took 17 years to get this project.
If it succeeds, sometimes never.
Yes, absolutely. High percent not succeeded. But
True. Great news.